So today we'll be quickly revising what is called as your biomolecules, right? So this chapter probably a little messed up chapter, right, where we had seen what are these particular molecules which are actually present inside the living cell, right? मग सगळ्या plants, animals आणि आपण काय म्हटलो होतो तुमचे मायक्रोब्स, राईट, या सगळ्यामध्ये जे ऑर्गॅनिझम्स मध्ये जे केमिकल कंपाऊंड्स प्रेझेंट आहेत या सगळ्यांना आपण काय म्हणतो तुमचे बायोमॉलिक्युल्स, राईट. Everything, if you analyze, that you have carbon, hydrogen, oxygen, and other elements which are present, right? All of them, they are present exactly in the similar way in the Earth's crust, right? So, if you are analyzing the Earth's crust, or if you are analyzing the human cell, or a plant cell, or an animal cell, or a microbial cell, most of the elemental composition ka same, right? And then you will find carbon, हायड्रोजन, ऑक्सिजन, नायट्रोजन, मे बी फॉस्फरस अँड सल्फर हे कुठं जास्त आहेत? दे आर प्रेझेंट मोर इन दी ह्युमन सेल, राईट. Whereas others like calcium, magnesium, sodium, potassium, they are more present in the Earth's crust, right? Silicon, they have mentioned, if you look at the table, it is present in a very negligible amount in the human cell, but it is present at a very high percentage. बघा किती percentage सांगितले for silicon. They are present, the silicon present in the Earth's crust किती? 27, right? आणि इथं आपल्यामध्ये कसा येतो? नेग्लिजिबल, राईट. सो क्वालिटेटिव्हली आपण दोघं कसे आहेत? सेम. वेदर वी आर टॉकिंग अबाउट अर्थ क्रस्ट or whether we are talking about human cell, qualitatively we are same, right? If you are analyzing these particular compound, if you are doing a proper analysis of these compound, we know that you do it for two things. One is organic, another is inorganic. For organic, what you call as analysis, what you're going to do, you're going to crush this particular tissue, right? And you're going to crush this tissue in trichloroacetic acid, and a TCA. TCA चा फॉर्म्युला काय दिलाय? CCl3COOH, है ना? कसं दिलंय फॉर्म्युला? CCl3COOH, right? So trichloroacetic acid, right? And this particular trichloroacetic acid you are going to put in mortar pestle, आणि मग mortar pestle मध्ये टाकल्यानंतर what you're going to do, you're going to make a paste of it, you're going to make a thick slurry out of it, आणि मग ती स्लरी तुम्ही काय करणार? फिल्टर किंवा स्ट्रेन. कशामधून फिल्टर किंवा स्ट्रेन करायला सांगितले त्यांनी? थ्रू दी चीज क्लॉथ, है ना? किंवा त्यांनी काय म्हटलंय? फ्रॉम द चीज क्लॉथ और कॉटन, right? And when you strain it, and you get two things. One is what is called as an acid insoluble fraction, and one fraction is called acid insoluble, and other fraction is what is called as the acid soluble, right? So all smaller molecules, they are in the acid soluble. This we also called as filtrate, you know, and this we call as retinate, right? So one which remains at the top, that is called as the retinate, and one which comes out, that is what is called as the filtrate, right? You have thousands of compounds, matlab nahi, thousands of molecules, thousands of ions which are present in the acid soluble fraction, right? मग आपण काय करणार? You can extract these particular compounds, you can separate it, right? It is all mixture. When you break open a cell, it is all mixture. So you can separate, you can purify, right? And once you have purified it, you have isolated it from all others, right? What you can do is you can also find out the chemical formula for it. म्हणजे त्याचा तुम्ही काय फाइंड आउट करू शकता? केमिकल फॉर्म्युला. But then how exactly it is done, that is beyond the scope of the book. But once you have these particular components, you can separate it out, and you can actually identify the compound, and you can have what you call as a purified fraction, and then you can also find out its chemical formula, right? So that can also be done, but again, as I told, that is beyond this thing. So all your carbon containing compounds which are there, which are present in the living cell. So how you define biomolecules? All the carbon containing compounds in the living cell, they are called as what? Biomolecules. So everything that is containing carbon and everything that is present in a living cell, right? That is what is called as a biomolecules. But then we know that apart from this, they also contain many inorganic elements, right? And these inorganic elements, they have told, you can perform a similar experiment, but which is more destructive. right? So you're going to actually destroy that particular thing. So you take the tissue, it may be your leaf, it may be your liver, right? And then first you dry it, right, to remove all the water, right? And then what you get is its dry weight, right? And then what you're going to do is you're going to actually burn it. तुम्ही त्याला काय करणार आहे? Burn. And once you burn it, right, what will happen? All the carbon compounds and organic compounds which are there, they will be converted to CO2 and water vapor. So carbon will get converted to CO2, hydrogen and oxygen will be converted to water, and what remains is all your elements, right? And then you can do elemental analysis where you can find that there are different elements. So here you will find different elements, like here you have, after burning, what remains is what is called as the ash, right? And then what you can find here is inorganic elements. You can find calcium, you can find magnesium, you can find compounds like sulfates, you can find compounds like phosphates, right? And you can find this carbon, hydrogen, oxygen, chlorine they have mentioned. So inorganic analysis for any compound, when you do, right, you will find all these components which are present, which are also found in the Earth's crust. So Earth's crust and your cell, they have both, right? For an organic chemist, right, if you're looking at the organic chemistry of it, people are interested in which functional groups are present, whether there is an aldehyde group, whether there is a ketonic group, whether the compound is aromatic, right? For what you call as biological chemist, right, or biochemist, what you call, you are interested in whether which organic molecules like amino acids are present, sugars are present, nucleotides are present, so which building blocks are present, right? A biochemist is interested in which amino acid or which sugar it is present, right? So that forms what is called as your biomolecule. So this is a rough analysis of these particular molecules, right? Now you look at molecule one by one. Now if you look at the first class of biomolecule which you have, that is what is called as your amino acids, right? So the first class of biomolecule konta hai tumsa amino acid, right? Which is the building block for protein. Amino acid is the building block for kai? Protein, right? Amino acid, we say that they are substituted kai methanes, right? Amino acids, they are said to be substituted methanes, right? So you have a carbon, then you have 4 valencies. Its 4 valencies satisfied by 4 different compounds, like you have hydrogen, then you have a NH2 group, a amino group, you have a carboxyl group, and you have a variable R group. So all amino acids are going to be different or based on only on their R group. Basic structure is going to remain the same. So what is different is only R. In nature, they are told there are how many different types? 20. So there are 20 different types of amino acids based on Konta group? R group. The amino and carboxyl are present to the same carbon, that is the alpha carbon, and therefore it is called as the alpha amino acid, right? So this particular amino acid which is there, that is an alpha amino acid. So as I told that what is your R group, that will decide the type of amino acid. So if your R is H, it is glycine. If your R is like CH3, that is methyl group, it is konta? Alanine. And if your R group is hydroxymethyl, then it is what is called as serine. So these 3 are given. Out of those all 20, they are given 3. One is like if your H is like R is H, it is glycine. If it is R is CH3, then it is alanine. If the R is hydroxymethyl, remember it is serine, right? So these 3 they have actually mentioned. So all the properties, remember, of these particular amino acid, they depend on the amino group, they depend on the carboxyl group, and also on the R group. So how this amino acid is going to react and how it is going to look like, or which reactions it will which will be taking part in, right? So all that will depend on the amino group, the carboxyl group, and the R group. So all 3 are actually what you call is going to define. Now number of your amino groups and number of your carboxyl group are going to define that whether it is going to be acidic, basic, or neutral. If it is having an extra amino group, it is basic, right? So they are given an example of lysine. You have lysine, arginine, histidine, which are basic. If it is having an extra carboxyl group, it is acidic. So you have glutamic acid, then you have your aspartic acid and all, which are what you call as acidic amino acid, right? If it is having equal number of them, so then it is what is called as neutral. The example which they have given is your valine, right? And if they are having a benzene ring or aromatic group in their structure, then they are said to be aromatic. Such amino acids are said to be kashe aromatic. Example konte konte dilet? Phenylalanine, then you have tyrosine, right? Then you have tryptophan, right? And then you have which one? Yes, the aromatic amino acid, phenylalanine, tyrosine, tryptophan, and अजून आहे कुठली aromatic आहे का कुठली? The aromatic amino acid, phenylalanine, tyrosine, and tryptophan. अजून आहे कुठली? नाही ना. So these are what you call as your aromatic amino acid, right? So your ionizable groups which are there. Now if I talk about your R group, now this R group, it is getting what you call as ionized here. If you see, this is the fully protonated form of the amino acid. What you have to remember is that if there is a change in pH, the ionization pattern of this changes. काय बदलल्यावर pH. So if I change the pH, this will go into the next phase, like the carboxyl group which is there, that will get ionized. If I go in the next phase, this will what you call as get deprotonated now. So here if I see the amino acid is having a net positive charge, here it is having a net zero charge, and here it is having a net negative charge. So here when both the groups are having charges, right, this particular form we call as which form? Zwitterion. This form is called as which form? Zwitterion. So Zwitterion form has both of them as charged, right? So this is protonated and that is deprotonated. So that particular form of amino acid is said to be the Zwitterion form. And remember that these charges on these amino acid, they depend on the pH. pH बदलला की charges पण काय होणार? Change, right? You join these amino acids by a peptide bond and you form what is called as a polypeptide. A long chain of amino acids, right? So you have one amino acid, second, third, fourth, like this. When you are joining many amino acids by a peptide bond, you form what is called as a polypeptide, or you form what is called as a protein. So there are 20 different types. I told there are 20 different types. Therefore, protein, remember, is said to be a heteropolymer. It is not a homopolymer. Like it's not only one amino acid which is joining. Any protein you take, it has a number of different types. It has a choice of how many? 20. So it is a heteropolymer. So entire protein is a heteropolymer of what is called as amino acids, right? So here you will find that these amino acids, again, they are classified as essential and non-essential, right? So remember, we have all 20, we require all 20, but those which cannot be made by our body. So you cannot make, your body cannot make that, and you're going to take it from the diet. So your diet is the source of those amino acids, then that is essential. The amino acid which can be synthesized in your body, remember you need all of them. So in the exam, if there is like whether you need a non-essential amino acid, yes you need it, but your body makes it. So you don't need it from the diet. Whereas essential amino acid I need from the diet because my body cannot make it. You'd be surprised to know that bacteria can make all 20 amino acids, but we humans cannot make, right? But that is a different story. So you have essential amino acid cannot be synthesized up by the body and therefore have to be taken from the diet. A non-essential amino acid can be synthesized in the body, therefore not required in the diet. NCERT does not give the list of those essential and non-essential amino acids, so I am omitting that. We are only following this NCERT. It's just like reading the NCERT line by line. Right, so we are going through only that, right? No concept, no explanation, right? So proteins, as you see that they are performing variety of functions. And proteins like polypeptide, when I said it is a protein, the proteins काय करतील? They are transporting, they are protecting, they are antibodies, they are catalyzing the reactions, they are enzymes, right? So proteins are like repair, wear and tear, build up, protection, maintenance of your body. हे सगळं काम कोण करणार आहे तुमचं? Proteins, right? Two proteins which you all remember always is your collagen and your rubisco, right? So collagen is like the one which is present, is most abundant in the animal world, whereas rubisco is most abundant in the biosphere. So in the entire biosphere, it is your rubisco, ribulose 1,5-bisphosphate carboxylase oxygenase, the first enzyme in which pathway? Calvin cycle, right? So that is your dark reaction. So that is the most abundant enzyme. It is the feeding enzyme. It has to be abundant because it is the enzyme which is taking that carbon dioxide from the atmosphere and giving it to you and to your entire world, right? So therefore it is the most abundant enzyme, whereas collagen, it is the most abundant protein in your konta world, animal world, right? For an inorganic chemist, remember they have told that the structure, it refers to your formula only. Like I say BSO4 or I say H2SO4, whatever like that. For inorganic chemist, that much is enough. For an organic chemist, they have told that they are interested in a two-dimensional structure. When they're looking at the structure of benzene, when they're looking at the structure of naphthalene, right? तसं त्यांनी सांगितलं. For a physicist, probably it is a three-dimensional view, like how exactly the structure looks like. So they are interested in the three-dimensional view. But for a biologist, and especially for a person who is studying protein, they categorize this into 4 levels of organization of protein. Kiti levels सांगितलेत? 4. So one is primary structure. Remember, primary structure is just a linear chain of amino acids. So amino acids are present in one chain where you can tell the position of each amino acid. मी सांगू शकतो, this is the first, this is not first, but this is the first, this is the fourth, this is the 10th, this is the 20th. But that straight chain of amino acid is said to be a primary structure. Remember, this primary structure will have on this side a N terminal and on this side it will have a C terminal. So every protein will have a N terminal and a C terminal. So this left side, the first amino acid which is there, it is having the N terminal. The last right side, it is having one terminal, C terminal. There are no structures given in your book related to protein or the peptide bonds, so I have skipped it, right? So here you have this primary structure, but as I said, that cannot remain linear as a rod. It will now fold on itself. So it will give a helical structure. So that forms what is called as a secondary structure, right? They have also shown another structure, but not mentioned there in the text, but in the diagram they have shown, if you see, there is a beta pleated sheet. And both they have labeled as secondary structure. एक त्यांनी काय दाखवले helix and दुसरे त्यांनी काय दाखवले beta pleated sheet. And both they have mentioned in कोणता structure, secondary structure. And they have told that this helix is like a revolving staircase. लक्षात घ्यायचं. And they have told that in the protein which are naturally occurring, all the helices, they are right-handed. So remember these small points here. Okay, proteins which are present in nature, they have told they have right-handed helical structure. And remember that there are other folded structures also, they have told, which is called as the secondary structure. So this protein may be folded on itself, parat sangitle, like a woolen ball, and that they have called as the tertiary structure. Remember, they have clearly mentioned that is this tertiary structure which is responsible for the 3D structure of the protein and is also requires for its activity. So which structure is important for its activity is konta structure? 3-dimensional structure. That is konta structure, that's a tertiary, right? Last, when more than one protein chain, it combines together. Like you have one protein chain, you have another protein chain, then another protein chain, then another protein chain, like 4 protein chains coming together. Example which they have given is of human hemoglobin, which contains 2 alpha and 2 beta, right? So these are a type of quaternary structure. They have called it as which structure? Quaternary. But they have mentioned like the linear structure of sphere or spheres on each other, right? In the form of a cube or a plate. Linear structure of spheres or spheres which are present on each other or in the form of cubes or plates, right? So that forms what is called as konta structure? Quaternary structure, right? So that is about your proteins. As I told that we are only going one by one, only what is given in your NCERT. It is just like reading an NCERT. So there is no explanation of the concept, or we are not going into the detailing of that, but then you need to remember each point. Then they have talked about another particular molecule which is called as lipids. One characteristic feature of lipids which they have told that they are insoluble. Water mein kaise wo? Insoluble, right? Whenever we are talking about repair, growth, wear and tear, it is your protein. But whenever we are talking about what you call as storage of energy, we are talking about lipids. Amino acids se khali dilela asil. The chapter is messed up a bit, so I took all amino acid proteins together. Now we'll be talking about lipids. So it is insoluble. It is also made of carbon, hydrogen, oxygen. Need not to mention proteins मध्ये nitrogen पण होतं है ना. Proteins मध्ये काय present होतं? Nitrogen. याच्यामध्ये it is not. So simplest, like they have told that it contains a fatty acid. Fatty acid is what? A hydrocarbon chain, right, which contains a COH. So you have a long hydrocarbon chain like CH3, CH2, CH2, CH2 आणि शेवटी काय? COH. Smallest methyl त्यांनी सांगितलंय, मग इथाइल त्यांनी सांगितलंय, and so on and so forth. Range कितना बोला है? Normally 1 to 19. And then two examples they are given. One is of palmitic acid, right, which is containing, including your carboxyl group, how many carbons? 16. Palmitic acid में कितने कार्बन है? 16. आणि दुसरा कोणता म्हटलंय? arachidonic acid. आणि arachidonic acid मध्ये किती कार्बन सांगितले? 20. So two examples they are given. One is palmitic, another is arachidonic. Palmitic is how many? 16. Arachidonic is how many? 20. Right. They may be saturated if they are having only single bonds, no double bonds, and therefore they are solids at room temperature, and therefore their melting point is also high. Right. They may be unsaturated, right, which have carbon, one or more carbon-carbon double bond or triple bonds. They are liquids at room temperature. The oils, example which they have given is of gingerly oil. You know, something they have given, which oil they have told? Check that. Gingerly oil, you know? And that gingerly oil they have told that it is liquid at room temperature, and obviously melting points are lower. So for fats, the melting points are higher. For oils, the melting points are lower. They contain saturated, they contain only single bonds, whereas unsaturated contain double and triple bonds, right? Some of them, they have told that they are what you call as combination of both. Some simple lipids, they have told they are combination of glycerol and fatty acid. And glycerol, they have told it is a trihydroxypropane. Right? So you remember trihydroxypropane. So propane matlab 3. Trihydroxy matlab it is going to have OH. So it is CH2OH, CHOH and CH2OH. So this is one molecule of glycerol. Plus 3 molecules of fatty acid. So you have 3 molecules of fatty acids here. So this will look like CH2O, C double bond OR1, then CHO, C double bond OR2, and CH2O, C double bond OR3. If it is only one fatty acid is esterified, then it is monoglyceride. If 2 are esterified, it is diglyceride, and with 3 are esterified, it is triglyceride. So all your fats, oils, they are generally triglycerides. Some compound lipids, they have told that they, instead of this third fatty acid, they will have a phosphate here. They will have what? Phosphate. And such are called as what? Phospholipids. And one example which they have told is that of lecithin, right? Where you have that CH3, then you have N, then you have those 3 CH3s attached to it, right? And that is what is called as the lecithin. And they have told that this lecithin is a phospholipid, and these phospholipids are mainly present in your cell membrane. Remember, lecithin kiwa phospholipids they put present in the cell membrane. Last, they have mentioned that some tissues, they have complex lipids. An example they have given of konta tissue? Neural tissues. They have given example of which tissue? Neural tissue. So neural tissues, they have some complex lipids. With God's grace, they have not given any structure of that complex lipid. They have not told which lipids are present in that, but they have sphingolipids and sphingomyelins and glycolipids and galactolipids. ते आता आपण नाही जाणार, पण फक्त लक्षात काय ठेवायचं की न्यूरल टिश्यू मध्ये कसे लिपिड्स आहेत? कॉम्प्लेक्स लिपिड्स. And as she told that you have those 4 fused ring like structure which we call as the cholesterol. आता ये cholesterol, they have just shown the diagram there, but they have not given in the text. So if you look at that one page of diagram, they have shown these 4 fused rings. I know these are not right, but now as I told that we are just having an overview. So it actually will look something like this, right? So this looks like what is called as cholesterol. So you remember we have dealt with this steroid nucleus when we were dealing with hormones like testosterone, estrogen, progesterone, even vitamin D. So they all are derived from this cholesterol. lipids like this is lipids like, right? But then these lipids, they create more complication in next sections if you read, because at one place they say that they are macromolecules, at the other place they say that they are not macromolecules. So you need to understand that lipids, they have clearly mentioned in NCERT, they are not strictly macromolecule. Two things to remember about lipids, that they are not strictly macromolecules. दोन गोष्टी, not strictly म्हणजे ते आहेत पण नाही पण आहेत. So they are not strictly. Another is that they are not polymeric. जसं we just saw amino acid join करून करून आपण काय बनवलं होतं, प्रोटीन. तसं असं खूप काय जॉईन करून करून आपण लिपिड नाही बनवलेलं. So they are not polymeric. So they are not polymeric, they are not strictly macromolecules. They have told that when we are talking about these particular lipids, when you are categorizing the molecules at acid soluble and insoluble, if you see, I am taking this now to the next level to not increase the confusion but to reduce it. But the acid soluble fraction has molecules which have a molecular range of 18 to 800. 18 to 800 असतील तर ते molecules कसे आहेत, micro का macro? Micro. If it is between 18 to 800. They have told that the insoluble fraction which is there, it contains 4 types which are macromolecular. And 4 types में कौन है आपके, polysaccharide which are carbohydrate, you have polypeptide or you have proteins, then you have nucleic acids, and then you also have lipids. इथं बाकीच्यांचा प्रॉब्लेम नाहीये, but प्रॉब्लेम कोणाचा आहे, फक्त lipids, because the lipids weight is less. इथं त्यांनी सांगितलंय molecular weight of these large macromolecules is more than 10,000 Daltons, remember. So try to remember here, 18 to 800 is the range for acid soluble. For most, they have told that it is about 10,000. But then again, in the next line, they will say that molecules which have a weight less than 1,000 also, they are micromolecules or biomolecules only. Whereas those which are present in the acid insoluble, and in the acid insoluble, if they are present, then they are macromolecules or biomacromolecules. So here they are defining it based on that weight. But here exception is कौन तुमचा, lipids, because the lipids weight is less than 800. But still they are in macromolecule, right? Why? Because they are arranged together, they are clustered together, they come together when they are present in the form of various aggregates in your membrane. So when you are opening the cell, you are breaking it. So various membranes are broken, the cell membrane is broken, the nuclear membrane is broken, other membranes are broken, but as soon as they are broken, they again cluster together. So because they cluster together, number one, and because they are insoluble in water, they come together, and therefore they appear in the macromolecular fraction. So they are broken into pieces and they will form various vesicles, and which are not soluble in water, as I told. And because they are not soluble, they will not go in the soluble fraction, they will come in the insoluble fraction. So therefore you have to understand that they are not strictly macromolecule, and also they are not polymeric. Another category which they have told that the acid soluble fraction, you have to remember this, it represents the cytoplasmic composition. Cytoplasmic composition, उन्होंने बोला है कौन सा fraction है, acid insoluble. And acid soluble and insoluble both, they represent the entire chemical composition. So remember these terms, sometimes they like mess you up with these terms, and then they give the proof of this NCERT. So you need to remember that if it is only acid soluble pool, they are saying that is konta composition, cytoplasmic. And if it is acid soluble plus insoluble plus those organelles, everything, then it represents the entire chemical composition. Right, so that is regarding what is called as your lipids and the confusion related to lipids, right? So if there is a question like they ask that which is the odd one, the polysaccharide dilet, nucleic acid dilet, then they have given your proteins and the lipids. In that case, it is lipids, because lipid is not strictly a macro molecule. But then it again varies. Suppose if they have given like find the odd man out and they have told lipids, then nucleic acid, then glycine, and then a polypeptide, then in that case it will be glycine. because though it is not strictly a macromolecule, it is still a macromolecule. Glycine surely is not a macromolecule because it is only one amino acid. मग कोणता कधी बाहेर काढायचा आणि कोणता कधी ऑड पकडायचा, that you have to understand. Then they have talked about another particular molecule, that is your nucleic acids. Now nucleic acid, we know that they are polymer of nucleotides. So as you join amino acid to form protein, you join nucleotides to form nucleic acid. Here they have mentioned that there are heterocyclic nitrogen containing compounds, and these heterocyclic nitrogen containing compounds they have talked about as nitrogen bases. And then if you talk about nitrogen base, then you have adenine, right? You have guanine, right? Then you have what? Cytosine. Then you have what? Uracil. And what is the last one? Thymine. Right? So these are the nitrogen bases. These also they are told that they are classified into two. This first two, they are purines, having only two rings. So adenine and guanine, they are said to be substituted purines. Cytosine, thymine, uracil, they are substituted pyrimidines. If you look at their structure, in purines you will find that there are two rings, whereas in pyrimidine you will find that there is a one ring. In that one page they have given the structure, please see that also. But if you want to differentiate between purine and pyrimidine, you can easily differentiate. If there are how many rings? Two rings, then it is purine. If there is one ring, it is pyrimidine. So thymine, cytosine, uracil is pyrimidine, adenine and guanine are purine. When this you are going to attach to a sugar, which is a pentose sugar, which may be ribose if it is RNA or deoxyribose if it is a DNA, any sugar. So then it becomes a nucleoside. So you have a nitrogen base, like I have a sugar. I'm not going to eat that, but and then I have a nitrogen base. I'm not going to detail of this because this is the part of next chapter in 12th standard. So we'll just have an overview कि एक nitrogen base एक glycosidic bond ने हा कुठे attach है तुमच्या sugar ला. It forms a nucleoside. So nucleoside is like adenine is called as adenosine. Guanine is called as guanosine. Cytosine is called as what? Cytidine. Remember, uracil is called as uridine and thymine is called as thymidine. So attachment of sugar, it converts them into nucleosides. If I attach a phosphate to it, this gets converted to nucleotide. So if I attach this phosphate to the fifth position, then it gets converted to a nucleotide. So remember, that's easy because that will be called as adenylic acid. Like this will be called as guanylic acid. So it's similarly cytidinic acid, uridinic acid, thymidinic acid. So it's open. So sometimes they have that odd one. So find out the odd one. So they'll give you adenosine, guanosine, cytosine, uridine, thymidine, all seen. So you have to understand that is cytosine, it is a nitrogen base, whereas all others are these nucleosides. So adenine, guanine, cytosine, uracil, thymine are nitrogen bases. Adenosine, guanosine, cytidine, uridine, thymidine are nucleosides. And all acids, adenylic, thymidinic, uridinic, cytidinic, they are nucleotides. So these are joined together and they form what is called as a nucleic acid, the two forms, the DNA and the RNA. And we know that they act as the genetic material. DNA acts as the genetic material in all organisms, almost all. RNA acts as genetic material only in certain viruses, right? Another thing to remember here, they have mentioned here, so I'll also mention that the difference in the sugar is at the 2' position. इथं मेन्शन केले त्यांनी की रायबोज मध्ये 2' पोझिशनला OH असतं आणि deoxyribose मध्ये it is only H. So they have mentioned, they have not like shown, but they have mentioned that it is deoxyribose at कोणता position? 2'. Right. So I think this is what they have just mentioned about what is called as nucleic acid. There are so many other things of DNA and RNA, but probably they are the part of the molecular base of inheritance. And as I told that we are focusing only on biomolecules chapter in NCERT one by one. Right. Then you have another class of macromolecules called as polysaccharides. You're also lucky they have not discussed monosaccharide, disaccharide. They've only discussed polysaccharide. So polysaccharide काय? They are polymers of monosaccharide, obviously. Polysaccharide कशाचे polymer आहेत? Monosaccharide. So many monosaccharide units joined by कोणती bond? Glycosidic. Try to remember those bonds. Huh? In protein it was peptide, in fats it was ester. You are lucky enough that in nucleic acid they didn't mention. But since you're going to give NEET on 5th May, you know that it is phosphodiester. So nucleotides, adjacent nucleotides, they are linked by phosphodiester linkage. And here the bond which is there, that is glycosidic. So many, along with your polypeptides, along with your polynucleotides, one more polymer which you have is your polysaccharides. Right? So these are long chain sugar which may have same or different monosaccharides. 2-3 examples which ideally you should not forget, which I get very angry when students forget, is these 3. The cellulose present in the plant cell wall, you're learning since kids. Starch, which is a storage product in plants, and glycogen, which is a storage product in animals. You have to remember here only that these all 3, they are made of glucose. So they are homopolysaccharides, but they are joined in a different way. Again, you all are lucky enough, they are not shown that in which way. Alpha bonds, beta 1,4 bond, beta 1,6 bond, nothing. काही नाही सांगितलेलं. एवढं तरी लक्षात ठेवा. Just remember that much की when many glucose joined in one pattern, it forms cellulose. When many glucose joined in another pattern, they form starch. When many glucose joined in another pattern, they form what is called as the glycogen. So these are your what is called as your polysaccharides. राईट. तर ग्लुकोज असेल, स्टार्च असेल, सेल्युलोज असेल, हे सगळे कसे प्रेझेंट आहेत? मतलब दे आर मेड ऑफ व्हॉट? ग्लुकोज. ऑल ऑफ दिस सेल्युलोज, स्टार्च अँड ग्लायकोजन. म्हणजे उलट पण विचारू शकतात, व्हॉट विल यू गेट व्हेन यू ब्रेक ग्लायकोजन? यू विल गेट ग्लुकोज. नाउ they have mentioned 2 ends. One is the right end and other is the left end. And they have mentioned that this right end is a reducing end and the left end is the non-reducing end. Since we are going only NCERT, no concepts, you just remember this. Right konta hai? Reducing. Left konta hai? Non-reducing. Bas. But then you know that those which have those free anomeric carbon, they are reducing, which can reduce them. And that's the concept मध्ये गेल्यावर वेळ जाईल. So right hand kashi reducing, left hand kashi non-reducing. Another which they have mentioned is inulin, which is a polymer of काय सांगितले? Fructose. Inulin कशाचा polymer आहे? Fructose. You join many fructose units and you get what is called as the inulin. Another thing which they have mentioned is starch is helical, and therefore who goes and sits in it? Iodine, and therefore it gives which color? Blue color. So starch can be detected with iodine. They have told cellulose does not have this helical structure, so iodine cannot fix and therefore it doesn't give any blue color. Helical structure कोणाचा आहे? Starch. त्याच्यात कोण बसतो? Iodine. म्हणून कोणता कलर? Blue color. Assertion reason. So this has a helical structure, it gives a blue color. So both are true and reason is the correct explanation for that assertion. Either cellulose will not give that blue color. Then they have told that all of them are not so similar, right? You have a cellulose example which they have told it is your paper pulp, paper pulp and your cotton fibers, right? So these are pure what is called as your cellulose. So they are nothing else but what is called as your cellulose. Some sugars are complex. Again, you are lucky enough they have not shown the structures of those complex. They have shown the structure of a ribose, of a glucose in that page. Please go through that. But they have not shown the structures of these complex sugars, which may contain amino sugars, right? Which contain chemically modified sugars like glucosamine, and they have told N-acetyl galactosamine. So it is glucosamine and N-acetyl galactosamine. And ek which they have asked twice or thrice in NEET is the exoskeleton of arthropods. Right, that is made of what is called as chitin. So remember, this chitin is a polysaccharide. Many people think that is a protein, chitin protein. It is not protein, it is polysaccharide. So it is a complex polysaccharide, and they have told that most complex polysaccharides, they are homopolymers, most of them. So most complex polysaccharides, they are kind homopolymers, right? Whereas others, they may be heteropolysaccharide when they are made of more than one, what you call as glucose units, right? Then you have the concept of what is called as a primary metabolite and a secondary metabolite. So you have to just remember that anything which is formed during metabolism, so you're degrading something, you're making something, and your small molecules are formed in between. Those particular molecules, they are called as metabolites. आप कुछ बना रहे, आप कुछ break कर रहे, उसमें जो बन रहा है, that is what is called as what? Metabolites. If its function is known, the role is identifiable. If you can find out, right, what function it is doing and what is its role, like it's a building block of what, then that is what is called as a primary metabolite. But with no identifiable role up till now, you have not discovered, it doesn't mean that doesn't have any role. But आतापर्यंत त्याचा काहीच रोल अजून आपल्याला कळालेला नाहीये, तर तो कसा झाला? Secondary metabolite. So they have told that this primary you will get in animals, and then secondary they have told you can get in plants, you can get in microbes, and fungi they are given separately. I don't know why, but fungi are part of microbes, but let it be. So thousands of compounds can be found in plant, microbial, and fungal. Some examples which they have given is alkaloid, flavonoids, rubbers, essential oils, antibiotics, colors, pigments, scents, gums, spices, right? So they do not have what you call as any role. At least we don't know at the moment what their role is. But they are useful to us. So rubber, scents, spices, right? We are using them, drugs. And then you will find that they also have ecological importance. Remember this. Sometimes I have seen many times that when you start reading a paragraph, all important things, and they ask question on the last line of the paragraph. So the last line says that it has ecological importance. So they are useful to us. We are using them as drugs, spices, scents, rubbers and all, but they are also having an ecological role. Nature मध्ये पण कोणते metabolites important आहेत? Secondary. Then they've given those charts. Those charts I'm going to take at the end, right? There are many charts in this chapter, but you need to know all those charts also. The last section now, right, which we'll be talking about is your enzymes, right? Now, if you talk about your enzymes, basically, all of them are almost all. So remember that almost all of them are proteins, not all. Some are nucleic acids. Nucleic acid acting as enzyme is called as ribozyme. In some other chapter, they have also mentioned which are those. Especially RNA molecules are acting as enzymes, and they are called as ribozymes. Enzymes, if you talk about, they are also having, like, they are proteins, as you told, almost all of them. So if you're looking at those proteins only, you will find that they also have a primary structure, a secondary structure, a tertiary structure, right? But they are active only when it is having a tertiary structure. And when it is having a tertiary structure, it is forming many crevices and pockets. And one crevice and pocket that is important is the active site. Active site is that part of the enzyme where what binds? Substrate. Active site is the site on the enzyme where what binds? Substrate. Remember. Enzyme, when we are talking about, they are biological catalyst, we say. Enzymes called as biological catalyst. Like we also have inorganic catalyst. But we know that inorganic catalyst can function at high temperature, they can function at high pressures. But these biological catalysts, they can function at normal temperature and normal pressure. You don't have to increase the temperatures and pressures. Whatever reactions are taking place inside your body, they are taking place in normal temperatures and pressures. Some enzymes can function at high temperature, right? Like those which you are getting enzymes from microbes which are living in sulfur vents, sulfur springs, hot vents, right? So from there, if you are getting, because there they are normally living at high temperatures around 80, 90, 100. So their optimum temperature will be above 80 also sometimes. So they are thermophilic. ऐसे organisms को हम क्या बोलते हैं? Thermophilic, which are working at कैसा temperature? High temperature. But otherwise they will die. Other organisms will die at that temperature. Normally 30, 40 is more than enough, right? Now here, when we are talking about a chemical reaction, when you are in your lower standard, you studied, if there is no formation or breaking of the bond, then that is a physical change. Change in the state of matter. Ice to water to vapor, physical change. Change in the shape, that is a physical change. But when a new bond is formed and the old bonds are broken, then that is kaun sa change? Chemical change. So they have given an example of a inorganic chemical reaction, like when your barium hydroxide is reacting with your sulfuric acid to produce this barium sulfate and water. Right, this forms a what you call as a chemical reaction, because you are actually breaking the bonds and you are making the bonds. So that is a consa reaction, chemical reaction. The rate of this reaction you can identify as change in the concentration of product per unit time. How much product is formed in the given period of time, that shows what is called as the change in the reaction. So that rate, it can also be called as velocity if you specify the direction. Like when I say that A is going to B, so that is what is called as velocity. One factor you need to understand, that for every 10 degree rise or decrease in the temperature, the rate of reaction will either double or the rate of reaction will become half. 10 degrees Celsius बढ़ गया temperature, rate of reaction double. If it decreases, it becomes half. For how much rise? 10 degree. If you have a normal reaction like CO2 combined with H2O to form H2CO3, carbonic acid, a reaction which normally takes place in your blood and your RBCs, catalyzed by carbonic anhydrase. They have told that if this reaction you allow to take place like that only without enzyme, how many molecules? 200 molecules per hour you will form. But if it is with the enzyme, you will form how many molecules? 6 lakh molecules per second. And you have to remember that this increases the rate how many times? 10 million times. So without enzyme it is 200 per hour. With enzyme it is 6 lakhs per second. And the rate has increased how many times? 10 million times. Remember, there are thousands of types of different reactions which are taking place inside the cell, and all are being catalyzed by your enzyme. Many reactions are multi-step. Like when we are converting glucose C6H12O6 to form your pyruvic acid. See this pyruvic acid formula, they are not given in that respiration, but they are given here. So remember this 2C3H4O3, that is your pyruvic acid. So it may be a multi-step reaction which may be catalyzed by many enzymes one by one. So that is also catalyzed by enzyme. Now what product is formed will depend on which enzymes are present and where the location is. Like if it is a skeletal muscle and if it is an anaerobic condition, then what you will form is lactic acid. Right? If it is yeast, then what you will form is ethanol in anaerobic condition. If it is an aerobic condition, then probably what you will form is your pyruvic acid, and this pyruvic acid will then be later on converted to CO2. So what is the end product will depend on what are the conditions and which is the organism. Organism बदलला, condition बदलले की rate of reaction पण काय होणार? Change. आणि rate म्हणण्यापेक्षा the products of the reaction काय होणार? Change. The molecule on which the enzyme act is called as the substrate, and what is formed is called as the product. Right? So enzyme is going to act on the substrate and it is going to form a ES complex, right? And it is going to form what is called as your product. So enzyme is going to bind substrate at the active site, right? And then you are going to form what is called as the product. So there is a compulsory formation of a ES complex, an obligatory formation of what is called as ES complex. And once the ES complex is formed, you have many unstable structures which are formed. So you have that popular graph of an enzyme catalyzed reaction where they show this energy. Here you have substrate, here you have product, and you have that graph, and then you show this. Now this is called as the transition state. So it goes through a high energy transient structure. Remember, it is a high energy state, number one, and it is transient. It is not permanent. The energy difference between your substrate and the transition state is called as activation energy. So here you can see this is the activation energy without enzyme, and this is the activation energy with enzyme. So with enzyme, the activation energy is less. So the rate is more. Activation energy कमी, alternate route आहे म्हणून activation energy कमी म्हणून rate is more. Without enzyme, sorry, when it is without enzyme, the activation energy is high and म्हणून वेळ कसा लागेल जास्त. So you will find that whether it is endothermic or exothermic, here you can see on y-axis we are plotting the energy, on x-axis you are plotting the progress of the reaction. Here you see that S is having more energy, P is having less energy. This is an exothermic reaction. If it is a reverse case where S is having more energy and S is having less energy, product is having more energy, then that is endothermic reaction. Whether it is exo, whether it is endo, they have told it is going to go in this transition state and it's going to require that activation energy. Clearly then they mentioned whether it is an exo or whether it is an endothermic reaction, it is going to go through these. आणि मग त्यामध्ये जाताना एन्झाईम ऑल्टरनेट रूट प्रोव्हाइड करणार आहे. म्हणजे असं काही नाही एक्झोथर्मिक मध्ये एन्झाईम एंडो मध्ये नाहीये, दोन्हीकडे काय एन्झाईम. दे हॅव गिवन द व्हेरियस स्टेप्स हाऊ इट हॅपन्स. सो दे हॅव टोल्ड दॅट एन्झाईम इट बाइंड्स द सबस्ट्रेट at the active site and it forms what is called as the ES complex. Now this what you call as there is a change, the steps they are given. There is a change in the active site to fit the substrate. The next step is that chemical bonds they are formed. Whatever, like you want to break it or you want to make it, and the last step is that your enzyme will be released and your product will be released. So that enzyme can do the reaction again. So these are 4 steps. Pehle enzyme will approach the substrate or substrate will approach the enzyme. It will bind at the active site. Active site will change. Bonds will be formed or broken. Enzyme will be released and product will also be released. So that enzyme can catalyze the reaction again. All this which is there, it can be affected by various factors, external factors. And some of the external factors which they have mentioned is what is called as temperature and pH. त्यांनी एक लाइन क्लियरली मेंशन केली आहे, ऑल दीज फॅक्टर ऑल्टर द टर्शियरी स्ट्रक्चर. रिमेंबर समटाइम्स दे विल आस्क दैट. इथं काय लिहिलंय, चेंज इन द कंडिशन्स ऑल्टर द टर्शियरी स्ट्रक्चर. सो यू आर इंटरेस्टेड इन दैट. टेम्परेचर आणि पीएच activity is in the narrow range. So if you look at the graphs of temperature and pH, they are like dumbbell shaped, right? Not dumbbell shaped, inverted bell shaped. Right. So there is a temperature and there is a pH where the activity is maximum and that is called as the optimum. You go on either side of the temperature or pH, the activity it decreases. What you have to remember here, low temperature preserves the enzyme. This is what you have to remember. Whereas high temperature it destroys the enzyme. It actually denatures it. You go to a high temperature, come back, रिएक्शन होणार नाही, कारण एन्झाईम परमनंटली काय झाली, डिनेचर. प्रोटीन्स गेट डिनेचर. Next, they have talked about the concentration of the substrate. They have clearly showed in the graph also that initially as the substrate concentration increases, the rate of reaction increases, reaches a maximum value. And that maximum is called as Vmax. A further increase in substrate does not increase the rate of reaction velocity. इथे त्यांनी काय म्हटले, अगेन दे हॅव शोन इन द डायग्राम बट नॉट इन द टेक्स्ट. If you go to Vmax by 2, the corresponding value of substrate concentration they have labeled as Km. In the channel you will find my entire video on how this is derived, what it means, but out of the scope of this book. इथं दाखवलं पण आहे, फक्त सांगितलं पण नाही. What you have to remember here is just it increases, then it remains constant. That maximum value is Vmax. That's what they have told. And why this happens? Because the enzyme becomes saturated with the substrate. Because the enzyme molecules are few and substrate molecules are more. All your enzyme molecules are now saturated with the substrate. So all they are saturated with the substrate. Therefore now you don't have any free enzyme to catalyze the reaction. म्हणून rate कसा राहतो? Constant. Assertion reason. And rate remains constant. Enzyme becomes saturated. Both are true. Reason is the explanation of the assertion. Right, that can happen. Then, as I told, that there are some other molecules which can stop the reaction, and those are called as inhibitors. The molecules which actually stop the reaction are called as inhibitors. Here you have to remember inhibitors which mimic the structure of substrate, which looks like the substrate, and compete with it to bind at the active site. They are called as competitive inhibitors. They are called as which inhibitor? Competitive. So competitive inhibitor example they have given is malonate, which looks like succinate, which inhibits the enzyme succinic dehydrogenase. So enzyme इथं लक्षात काय ठेवायचं? एन्झाईम सक्सिनिक डिहायड्रोजनेज आहे. मॅलोनेट हा कोण आहे? इन्हिबिटर. आणि सक्सिनेट हा कोण आहे? सबस्ट्रेट. मग मॅलोनेट कोणासारखा दिसतो? सक्सिनेट. Right, so malonate looks like succinate and stops this succinic dehydrogenase. Last line again. Focus on that last line. काय लिहिलंय? It is used in control of bacterial pathogens. So these inhibitors, they are used in control of what? Bacterial pathogens. So there are many drugs which will stop the activity of your enzyme of bacteria. Don't stop your activity. We want to kill bacteria. We are already killing ourselves by studying all this. So talking about what is called as your enzymes, you classify the enzymes, nomenclate the enzymes based on their reactions. Remember, enzymes are not known by who they are. Enzymes are known by what they do. You also will be known by what you do. So enzymes are known by what reaction they catalyze. And there are how many classes? 6 classes, right? And they are told that every enzyme has how many digit number? 4 digit. Every enzyme is known by how many digit number? 4 digit number. So there are 6 classes and every class is around 4 to 13 subclasses. This they can ask in which of the following statements are correct. So the enzymes are known by their structure. Enzymes are known by the reaction they catalyze. Enzymes have 6 classes. Enzyme have 4 to 13 subclasses, and every enzyme has 4 digit numbers. So only that first one was wrong. They are not known by their structure, but they are known by their function. 6 classes में पहला कौन सा बोला उन्होंने? ऑक्सीडो रिडक्टेज. नाम में ये which catalyze oxidation and reduction. So removing and adding of hydrogen, removing and addition of oxygen, or removing or addition of electrons. So you have something which is reduced, you have something which is oxidized. So the one which is reduced will become oxidized, the one which is oxidized will become reduced. So oxidoreductases, also called as dehydrogenases. Then second is transferase. Transfer any other group. I don't know why they love this G, maybe because my name is Girish. But then any other group other than G. Oh shit. Any other group other than hydrogen. If that is being transferred. So if this S has G, now this goes to this. Oh, both are not having. So this S gives it to this. So now you transfer this to this. So that is transferase. Then it is your hydrolase. Hydrolase naam hi bata raha hai. It catalyzes hydrolysis. So breaking of the bonds by addition of water. Right. So that is what is called as hydro. They are given the list of the bonds. Oh my God. Ester, ether, peptide, glycosides, C, C halide and PN bonds. Then they have talked about lyases, which are going to break the bonds, add a double bond with mechanism other than hydrolysis, without water. So here they have shown this introduction of double bonds. They have broken these bonds. So that is your lyase. And then they have talked about isomerases, which catalyzes the interconversion of isomers. 3 types of isomers they have mentioned: geometric, positional, and optical. And then last they have told is ligase. Ligase, what is going to do? Joining. Fevicol ka mazboot jod. So it is going to join the molecules and form new bonds. That is what is called as ligases. Bonds, now they are given the bonds. CO bond, CS bond, CN bond, PO bond. So different bonds it is forming. Enzymes, like all of them, they don't have only protein part. Some they have a non-protein part. And that non-protein part of the enzyme they are called as the cofactors. So all the non-protein part they are called as what? Cofactors. So cofactors, the protein part now is called as the apoenzyme. So remember, the protein part of the enzyme is the apoenzyme, whereas the non-protein part which is there, that is the cofactor. This they have told is 3. One is prosthetic group, other is a coenzyme, and third is the metal ion. So prosthetic group is tightly bound, cannot be removed. एग्जाम्पल कौन सा ग्रुप दिया उन्होंने? हीम ग्रुप, इन विच एंजाइम peroxidase and catalase, which are actually catalyzing the hydrolysis of hydrogen peroxide to form H2O and O2. Coenzyme is loosely bound, transiently bound, generally bound when the reaction is taking place. They can be part of different enzymatic reactions also. Most of them they have told are vitamins. Example, they have told us nicotinamide adenine dinucleotide. Oh my god, NAD and NADP. And the vitamin which is present in this is niacin. And then metal ions, they are forming coordination bonds. So on one side they are attaching the active site, on the other side they are attaching with the substrate. So they can bring these together in the close proximity. So they have a side chain attaching the active site and coordination bond with the substrate. Example they are given of zinc, which acts as the cofactor in carboxypeptidase, which is a proteolytic enzyme, an enzyme which hydrolyzes proteins. Hey Dhoni, prosthetic group and coenzyme. One common factor in this is that they are organic. But they can tell now what are common, what are uncommon. So prosthetic coenzyme, both are a type of cofactor, common. Both are organic, common. Both are important for catalyzing the reaction, common. Both are tightly bound, not common. Who is only tightly bound? Prosthetic group. And remember, last line again, as I told, last line, catalytic activity is lost if your cofactor is removed. So if you remove the cofactor, the catalytic activity is lost. So they can tell like statement 1, catalytic activity is lost if the cofactor is removed. Then next, cofactor is very essential for the catalytic activity of the enzyme. Does statement 2 prove statement 1? The answer is yes. Because if you remove the cofactor, catalytic activity is lost. It only proves that the catalytic activity is because of these cofactors. So in almost an hour, we finished with this entire what you call as chapter of this biomolecules. You have separate videos on the channel, like cofactor will separate video of 20 minutes. So different things are there. So tomorrow we'll again come up with a new chapter, right? Thank you. What am I supposed to do now?